This is episode 301 of the Stem Cell Podcast, Cell-Fate Decisions in Cancer, with Dr. Mark Hatley. Hello, everybody. We are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying the listen, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today on the show, we have Dr. Mark Hatley, who's from St. Jude Children's Research Hospital, and he's on the podcast to talk about his research on cell-fate decisions in pediatric cancer. We've also got our usual roundup of recent highlights and stem cell news that's coming right up.
But first, this week, we'd like to remind our listeners about Cancer Stem Cell News, one of Stem Cell's free bi-weekly scientific newsletters. Cancer Stem Cell News summarizes all of the latest research, news, jobs, and events in cancer stem cell research and delivers it right to your inbox every other Wednesday. Save time and keep current with Cancer Stem Cell News. Subscribe for free at cancerstemcellnews.com. We're going to start things off with something really just dropped in the New England Journal of Medicine, of all places. And if it's a New England Journal of Medicine paper, you know it's going to be clinically oriented and it's going to make a big splash, which is kind of what this is. And we've known about this for a while. This is building off of the work of Sonia Schrepfer and the folks at Sana Biotherapeutics and UCSF as well. If we're mentioning Sonia Schrepfer, we're talking about hypoimmune iPSCs, hypoimmune pluripotent stem cells, and hypoimmune cells in general. And that's the deal here. So they've basically, and the title of the paper kind of summarizes it, survival of transplanted allogeneic beta cells with no immunosuppression. And that's the key right there, no immunosuppression. So we're talking about type 1 diabetes, right? And we know that there's a need to suppress a patient's immune system after transplantation of allogeneic cells. That the whole immunosuppressive process is really risky. And we've talked about this at length with Dr. Honghui Deng over at the ISSCR this year, and he's got his own approach to treating type 1 diabetes with these chemical iPSCs. And in fact, we're going to have a paper later up in the Roundup that's really focused on that as well. But here they're generating these allogeneic cells with hypoimmune modifications to treat type 1 diabetes. Again, really the major focus of the work at Sana, right? And this is showing the outcomes of these genetically modified allogeneic donor islet cells into a guy with long-standing type 1 diabetes. So this is not IPS, right? This is modifying primary allogeneic donor islet cells into somebody with type 1 diabetes. Then they use CRISPR. So they use CRISPR to modify via lentiviral transduction and edit these cells to actually avoid the rejection, right? So they had all these edits for hypoimmune evasion. And then the cells were actually transplanted into the participant's forearm. You don't have to put them into the pancreas. That's risky. You could just put them into the forearm where the cells are actually just connected to the circulatory system and just doing their job and secreting insulin and regulating glucose, all that kind of stuff. And importantly, thanks to those hypoimmune edits, this individual did not receive any immunosuppressive drugs, and at 12 weeks after the transplantation showed no immune response against those gene-edited cells. This is, again, allogeneic donor, but no immune response thanks to those hypoimmune edits. Pretty groundbreaking, incredible stuff here. And then you got to validate that the cells that are being transplanted are actually doing their job, right? So the C-peptide measurements were able to show stable glucose-responsive insulin secretion. And there were 4 adverse events that were reported that they just, laid out here just to be transparent. But apparently none of these were serious or related to the study drug. So this is N of 1. Sure, N of 1, and really just groundbreaking N of 1, I think, for this publication in New England Journal, right? This is groundbreaking in a number of ways. Of course, you have the use of the allogeneic donor cells for treating type 1D, but it's the hypoimmune component, right? The modification of these donor cells via CRISPR, Cas12b editing in particular, to shield the host immune system from donor cells and prevent the creation of the immune response. So much more work to be done here, but really a milestone, a landmark, whatever you want to call it. We've seen this data presented in various contexts. And in fact, full disclosure, Sonia Schrepfer is actually on the faculty here at Cedars-Sinai in some part-time. Also, she's at UCSF. So we've had these conversations. We're all very excited about her work here and potentially future applications, right? That's the inevitable next step. Is like, you got this system for hypoimmune modification. Can you use that for other sources, other situations of cell therapy? I think that's the inevitable next step here.
This is a crazy study. I mean, it's not crazy. This has been a long time coming, and it's totally reasonable and fair. The approach, everything was well designed and done with deliberate care. But this is the realization of everything we've been dreaming and talking about. You ask, N equals 1 for a pretty early endpoint in terms, it's not a cure, just function, survival. Why is this out in NEJM of all places? Well, because this is, as you said, the moment. This is like the watershed, hits all the markers, and specifically the hypoimmune and the survival and the function. And, in light of a few things, one, I think it's particularly important that this is out in light of, I'm about to talk about some, I don't want to say negative results, but a bit of a reality check in terms of the whole xenotransplantation model with pig sourced material. Also the Vertex, Vertex cured diabetes, or the therapy at Vertex now for diabetes is an effective cure, but only with immune suppression, right? And the trial that was using these kind of rods, they arrested because it wasn't working, mostly because of like a acute fibrotic response to the actual implant device itself, which was presumably really watering down or interfering with the communication between the cells within the transplant and the endocrine system. So here, this is a watershed moment. And I think this is the key to the success here, is that, I don't know, I could be wrong. I got to look into the details a bit more, like you said, or in this just dropped, but they inject these cells directly, as I'm looking with like a pearl on a string distribution. They just inject directly into the incision and then pull back the needle with a lot of injections, 80 million cells prepared in 17 syringes, 17 injections, but just directly, as opposed to using an apparatus or some kind of scaffold, which may introduce another layer of complication with efficacy. So I think this is great. My curiosity is multifold, of course, but the first question I'd wonder is, with the long-term endpoints here, are they expanding this to multiple patients? I think the rationale for doing that is clearly there. And more interested in, ultimately, how are these cells going to be administered? Like, and we talked to Hunky Dang, I remember you're talking about the rectus sheath, if I have it right, which was this unique site which got around the whole portal vein and the flux there and retention of the graft. But here, like, is this the modus here? You go into just a site, just the cells. Do you have to secure them there? Do you just put them into the space and don't think about them? Is this perhaps a model for other cell therapies that are more modular, where you don't need the whole organ, like maybe liver or like hormone replacement therapy, which I'm interested in with the cell-based thing? Can you just put the cells in? Can you do it without a scaffold? So many questions here, but ultimately this is just really just a tremendous validation. And I'm just so excited at the series of studies that are sure to come with these hypoimmune cells.
Yeah, for sure. I think many things to be excited about here. Of course, the hypoimmune part of it and the accessibility of the therapy, like what you're alluding to here. This is an intramuscular injection, right? I don't know if there's like local anesthesia or probably, right? But, you can think about how you would be able to extend this approach, just like what you're saying with like hepatocytes, liver, anything that needs to be connected to the circulatory system in this situation, these insulin secreting cells. Long-term stability, long-term survival, and all these endpoints are needed, of course, but very exciting nonetheless. And on a bigger scale, when you're talking about just type 1 diabetes, so many exciting shots on goal right now for the stem cell field when it comes to treating, potentially curing type 1D, which is of course a medical scourge across the world, whether it's, injection of chemical iPS derived pancreatic beta cells into the abdominal rectus sheath, like you're alluding to, or intramuscular injection of these modified allogeneic donor cells. And the limitation here is, of course, in the donor cell part of it, that cell sourcing is not unlimited in contrast to what you would get with iPS. So perhaps that is a restriction here, but nonetheless, really powerful technology.
Yeah, I mean, and off the shelf. I mean, forget it. This is it. But as you're saying there, this is a lot of shots on goal and the culmination of many years of therapy. And ultimately, what's great about this, and this is a segue to my next story, is that how it's like kind of I don't want to call it a hack, but you have a lot of incremental progress, and then you have these breakthroughs, right? Like the whole approach was, okay, yes, we got to make the cells, and iPS, we can get patient specific, and that was really, full steam ahead. But now this hypoimmune addresses so many issues, and creates a product that really is scalable and can be applied across a breadth of diseases. So yeah, this is such an exciting start to this clinical translational phase of the science. And as I said, sometimes it's like a jump forward, right? And that's certainly the case with the whole xenotransplantation of whole organs, right? The idea there being, who needs to make an organ, right? That seems like a super heavy lift. There's not enough organs for transplantation. We'll get organs from pigs. We got infinity pigs. We eat a lot of pig. And that, talk about amazing progress with the heart, with the kidney, with the liver even. And that's a story I'm going to talk about. There was a previous study that transplanted genetically these hypoimmune, non-rejectable, genetically modified pigs where their organs are transplanted into humans. There was a previous study done by the same group, which is centered out of a Zhejiang hospital in Xi'an, China. This story led by Lin Wang, also Keifeng Du and Deke Pan. Sorry for the mispronunciations, but this is a big group who had previously demonstrated that they had transplanted one of these xeno pig livers into a brain dead human. And this was a really encouraging study. Like many of these, there was no sign of organ rejection after 10 days of transplantation. It was widely considered to be an early success story. But when you look closer at these, this specific case, the serum bilirubin levels progressively increase in the middle to the late stage and phase of this transplantation. So they thought this was a kind of dysfunction, or what they describe as a metabolic dysfunction of the transplanted liver. The hypothesis there was that this may be accounted for by this immunological response. And this is in spite of the fact that they looked at the liver, of course, and showed there was still regenerative. It showed that you could get like Ki-67 proliferation, regeneration of that pig liver. Also, no sign of fibrogenesis, only slight sinusoidal congestion. So the sinusoidal vessels, no blockages, suggesting like vascular dysfunction or thrombosis, and no sign of inflammatory cell infiltration, as well as lack of any of these inflammatory indicators like C-reactive protein, right? But there's been other studies, and most notably a study we talked about on the podcast, as well as there's other studies of the same, I think it was xenotransplanted kidneys, showing that there was kind of this innate evidence of this innate immune response, specifically like in the capillaries of the transplanted organ. And in those studies, they undertook this longitudinal transcriptomic analysis of the blood in the patients of the peripheral blood, also looking at the kidneys. So in a similar vein, this group performed single cell RNA-seq also of this one patient, brain dead patient who had the liver in there for 10 days before they took it out. They did a lot of recovery of the patient blood at multiple time points, 2 hours, 1 day, 3 day, 5 days, 8 days, and 10 days after transplantation. So these longitudinal time points taking peripheral blood. They also did spatial seq on the pig liver. And this is the content of this story. Another N equals 1. And just even a follow-up, kind of 2-for-1 of a previous story that was already highlighted, but here doing the postmortem, literally. And this is a Nature Medicine story. So a lot of parallels here with the NEJM. When you start getting these really clinical medical journals, scientific, but really clinical, talking about this stuff, we're getting close to prime time. And here, this is a postmortem to see what are the obstacles. And I think there's a lot more caution. While I feel the hypoimmune stuff is really ready for prime time, next 10 years, I feel like therapies will be in play off the shelf. The xeno might run into some obstacles due to this, these unknown unknowns. And what they found here really illuminated some of those unknowns, which I think ultimately is going to be the solution to find what the obstacles are to success in addressing them in a systematic way. And what they found here is that there was a lot of T cells that were progressively activated in the peripheral blood of this single patient. And those T cells infiltrated the pig liver extensively, showing that there was impaired adaptive immunity in this case. And what they found specifically is that there were 2 waves of monocytes. First, this thrombospondin positive monocyte that they postulated might be regulating coagulation or resulting in some kind of like thrombotic interference. And then second, another phase that promoted T cell exhaustion, another phase of these monocytes that infiltrated the pig liver and promoting T cell exhaustion. So here are 2 basic observations that are really elevated by the context here, the xenotransplanted patient, which was a success, but with challenges related to the innate immune cells, and specifically here, how both thrombotic and immune dysregulation might interfere with the success of these transplanted organs, their function and long-term retention. And as I mentioned earlier, the key advance here being is by identifying those 2 cells and identifying the phases that are interfering with success. I feel like these are targets, right? That you could maybe mitigate the negative interference there during this transient phase, acute phase following transplantation, and maybe rescue these organs or ensure a better outcome in these patients. So, like, I don't know, I don't want to call it a negative result because all these were so early days with the xenotransplantation studies. I think it's really important to identify and be clear about what the challenges are, because unlike these hypoimmune cells where there's a fallback plan, there's a backstop, both therapeutically as well as cellular, you can add more cells. The idea of serial pig organs being transplanted into the patient, while that may work as a stopgap while you're on the waiting list for a true human organ, I think we're not really ready to move forward with the xenotransplantation in practice until we're very clear on what the long-term function and challenges to long-term function are.
Yeah, but just think how far we've come even in the last 5 years.
Sure.
Like
Absolutely.
you pick your major organ, heart, kidney, liver. Now it's happening. Like, these things are being put into people, and again, N of 1, very early phase trials. But the fact that we're able to even get to this point is just miraculous in itself. Not miraculous. It's built on decades of work. it all kind of comes down to our friend or our enemy, the immune system. I suppose that's the universal issue for all these studies. And no, we're not the immunology podcast. We are the stem cell podcast, but it seems like every single cell therapy paper that we're ever going to cover is, of course, going to have to address this problem of the immune system one way or another. You pick the application.
Yeah. And like you said, what an amazing leap forward. The fact that we're talking about what the challenges to long-term success, just taking it for granted that we've got these organs that are surviving and functioning for all intents and purposes in humans. So yeah, a hack, like I said earlier, sometimes you have these big leaps forward, and this field is certainly one of them on two sides of it here, talking about both the hypoimmune cellular component as well as the whole organ, pig organ, xenotransplantation. Very exciting time.
Absolutely. And of course, 20 years or so ago, the gigantic leap in the stem cell field was the production of iPS. And in fact, next year we have that amazing meeting that's going to happen in Kyoto celebrating the 20-year anniversary of the derivation of iPS by Yamanaka and colleagues. And in the spirit of that, the third paper we're going to talk about here on the Roundup today is a cell stem cell paper focused on reprogramming, of all things. We're still talking about reprogramming studies here in 2025. But the reason this is a cell stem cell paper is that it's a special type of reprogramming. The work that's, of course, led by Honghui Deng, who we interviewed at ICCR 2025 in Hong Kong. This is chemical reprogramming. And he's demonstrated that, you can do it and you can use it to make iPSCs into pancreatic beta cells or whatever that can then be used for clinical trials. That's already happening. And that was the focus of our conversation with him and Candice Liu as well. But there are still certain limitations with the process, right? Namely that it's been largely done in fibroblasts. Now, the gold standard for making iPSCs these days, using the old school quote methods, are using PBMCs, like peripheral blood cells, right? And until now, they haven't been able to show in the Deng lab and associated labs that you can use this chemical reprogramming approach for turning human blood into pluripotent stem cells. And so that's what they did here. They demonstrated a robust chemical reprogramming method for human blood cells. We've talked about chemical reprogramming in the past. Of course, we had the interview. You should check out that episode as well at ICCR. But the major advantage of chemical reprogramming is that it's perhaps a cleaner approach than traditional approaches that are using genetic overexpression, ascendovirus, all this kind of stuff. You're just using small molecules, right, to make iPSCs. They have shown throughout the years that this is a really efficient process in fibroblasts, turning human fibroblasts into human chemical iPS cells. But doing it from blood, reprogramming from peripheral blood, has been challenging. And like I said at the top, peripheral blood is the most accessible cell type for reprogramming. Nobody wants a punch biopsy, right? I don't want a punch biopsy. I'd rather just get a blood draw. So here they created this robust method for generating chemical iPS cells, human, from both cord blood and also from adult peripheral blood, which is predominantly the source for iPS generation. It was an efficient process, but in both fresh, cryopreserved samples, which is great because you want to be able to ship PBMCs across the world for programming, and created an average of over 100 different chemical iPS colonies from just one drop of fingerstick blood. So you no longer have to get like 10 mils of blood for reprogramming. You could just generate iPSCs from a drop of blood. And so there's I think immediate applications here. They describe the kind of the workflow, the process, the characterization of the iPSCs, all the usual stuff you would do for iPS generation. It's again, cocktail of small molecules, summary of the small molecule. You can just look at the paper. It's a programming paper. It's, you're characterizing iPSCs, if nothing else, but it's establishing the workflow. But there's still some stuff that they have to do to actually really systematically assess that the iPSCs that you get from this approach are the same quality as you would get from other approaches, abnormal imprinting, genetic mutations, all these kind of things have to be characterized in future studies. But it's a protocol, and it's potentially a very, very powerful one at that.
Yeah, this kind of stuff you said at this point in the game maybe sounds like a little bit boilerplate, but it's important. And the efficiency key here, you know, number single drop of blood, high efficiency, soup to nuts in terms of the SOPs. But for me, what immediately jumps to mind is what everybody asks me when they hear I'm a cell kind of scientist or in reproduction. I don't know why, but they always ask me, oh, should I save my kid's cord blood? And I always say yes. And here's the answer right here, right? I mean, if you got your cord blood stored, and we talk about this a little bit, not enough, about, my skin cells are banged up after all these years, and blood with chip, with clonal hematopoiesis, indeterminate potential. Do we really want our own peripheral blood mononuclear cells to be the cell of origin of a cell therapy that we may have to have in our bodies for some time? I don't know, but that cord blood is fresh and clean, unblemished, unadulterated. So if you did save the cord blood of your kid, you're in business. I mean, we used to think it was just about insurance policy for a few kind of hematologic malignancies, but here we are now. It's the fountain of youth. So I donated mine. So sorry, kiddos, but Arun, I hope you saved your boys.
I did not, but hey, maybe they're still little.
So draw their blood now. They're still young. They haven't been tainted, even with all that LA wildfire smog. They're still clean.
Fair enough. We got 6-month-old. We got 5-year-old. So they're still on the younger side of things. So, hey, yeah, absolutely nice application for cord blood there for iPS reprogramming, but bigger picture, I think this is just going to open the door further for some of the downstream work that's coming from the Deng Lab and the associate companies spun out from the chemical reprogramming work. So, the writing on the wall is obvious here. They're going to use, if they can show that this works for, blood, this chemical reprogramming approach, they can, of course, use it as their workhorse here. Nobody wants a punch biopsy. Like I said, I don't want a punch biopsy. Those things leave scars. So finger prick, no problem.
It's the future. Less is more. We're staying in the blood and also talking a little bit about immunity here, kind of immunity adjacent. Sorry to the immunology podcast if we're drinking your milkshake a little bit today. The story about Notch, and you talked a bit about iPSCs. People forget, but George Daly, he was, right there, neck and neck with Shinya Yamanaka, and the first to demonstrate iPSCs in human. Of course, Shinya deserves all the credit, and Daly was using his recipe, but still. Point being is he's a early adopter, and he's been grinding across the board in regenerative medicine, stem cells, pluripotent cells, but really his first love has always been the blood. And Notch, really, he's had many, many stories that focused on Notch signaling. World expert in Notch signaling because it's been the thorn in his side, you might argue, and the thorn on the side of many hematologists that are trying to gain some mastery over differentiation and training of hematopoietic cells for therapy. So yeah, a key factor about Notch, and this is a bit the challenge, it's a key developmental fate specifier in many contexts, a lot in the blood, but you need direct contact between the sender cells, that present the ligands like DLL, delta-like, or jagged, and the receiving cells that have the receptor, which are the Notch 1 through 4 receptors. And you actually don't just need them to be membrane anchored, but you need a mechanical force to be exerted by that ligand, the delta-like or jagged. And that's what ultimately liberates this Notch intracellular domain, which goes down to the nucleus and drives downstream transcription. So in order to hack that in the past, like most ligands that we add, the receptors express, we dump the ligands in, they're soluble, bang. But with Notch, you got to jump through all these hoops, right? You got to present it, the ligand, in like a transmembrane fashion, or immobilize it, like on tissue culture disk surface or on microbeads or something. But yeah, like I said, Notch, it's involved in so many things, but really most notably, therapeutically, it's involved in maturation of T cells and enhancement of their effector function. And that can be really key for a lot of biology, but clinically, therapeutically, it's key for T cell manufacture, right? Also enhancing vaccine efficacy, which is another important endpoint. But now in the era of like, adaptive immune mobilization of these adaptive immune therapies, T cell manufacture is a big target. And also, if you want to differentiate human-induced pluripotent stem cells to T cells for this kind of adoptive cell therapy, or other approaches like immune deficiency, or treatment of anything autoimmunity, cancer, you want to have like an off-the-shelf thing, right? Like we were talking about the hypoimmunes. But if you want to generate this golden cell, T cell or any hematopoietic cell, you need a lot of Notch signal modulation, right? So, as I said, in the past, Daly, as well as many others, have used like a stromal cell that expresses Notch ligands, or you anchor it to microbeads or tissue culture plates. But here, Daly Group, this is a culmination of many, many years of work and a ton of human power. They hit upon this soluble Notch ligand by taking this computational protein design. Again, culmination of so many years, and now we are here in this kind of AI universe where AlphaFold and similar programs can actually undertake intelligent design de novo of proteins. In this case, using this Rosetta protein design tool. There's a lot of chemistry here and a lot of validation. But bottom line is they developed this soluble Notch receptor agonists that promote cell bridging. They cluster the Notch receptors at cell synapses, and they activate receptor signaling using these reporters that they have. They validate. Also, functionally speaking, they drive T cell differentiation from cord blood. There's cord blood again. So hope you banked it for your kiddos. And also key in human-induced pluripotent stem cells, they foster T cell differentiation from those. And in liquid suspension using bioreactor, they are able to produce T cells like at scale. Also, when they put them into mice delivered IV, they stimulated cytokine production and expansion of these CD4 positive T cells and antibody class switching, all validations of these hallmarks of T cell function. Of course, there's a lot of story to be told from this point. This is a Cell article, because just all the chemistry and design that went into generating these things. And talk about impact. Everybody's going to want a piece of these. But the story that has yet to be told is what I can't wait to hear about, which is applying this mastery of differentiation toward therapeutic endpoints, of which there are really too many to talk about right here. It's just the whole world. Not all the problems, but so many problems could be solved by having mastery over hematopoietic and specifically T cell differentiation. So a big and impactful story from the Daly Lab. Arun, what do you think?
Yeah, Daly Lab's been doing it longer than pretty much anybody, right? They are one of the preeminent stem cell labs in the whole world. And of course, Dr. Daly was the head of the Harvard Medical School for a little while there. I actually don't know. Maybe is he still the head of the Harvard Medical School? But either way, his lab is still hella productive. We're talking about Notch, of course, one of these, critical signaling pathways, Notch delta. It's so critical for T cells and, cancer and all these different applications. It's right up there with Wnt. It's one of the ubiquitous pathways out there. Really exciting study. I think one thing to focus in on, and they mentioned this in the limitations, is that certainly there's like a diversity of Notch receptors, and they have all different types of expression across different cell types, different binding affinity, cross inhibition, cis inhibition, like all this kind of stuff. And in particular, while this DLL4 is activating all Notch receptors, you gotta, there's a ton of applications out there that you have to really finely focus and specifically activate a particular Notch receptor paralog, right? So B cell development in the spleen, like this is just stuff that they reference. Like spleen specifically requires activation of Notch2. Okay, so that's going to be the inevitable future study here, is to figure out strategies that can have selective activation of single Notch receptors in specific contexts to really flesh out these functional roles. But it's a daily lab. They've probably already done that.
Daily lab, doing it on the daily. Yeah, and he is indeed dean of Harvard Medical School. And yeah, you said it there. Like, there's a whole story yet to be told I'm talking about in terms of the therapeutic impact. But this is the other story yet to be told, which is all the biology in Notch. And this is what has plagued anyone who's tried to work with the pathway, right? Unlike Wnt, which is, I mean, ubiquitous and pretty, dumb, for lack of a better word. You throw on the Wnt and activate, you can inhibit it, you can activate it with chemicals, with ligands, basic. But Notch, a lot of times you're doing the assay, having worked with Notch, you'll be doing the assay and you'll be like, oh man, I just added, I think I added my Notch ligand, but it wasn't anchored correctly, perhaps. Right. So instead of exerting that force that's necessary, the mechanical force that's necessary for signal transduction, I might have been inhibiting it. So like there's all these assays that you do where you're like, OK, I'm adding Notch. No, I'm inhibiting Notch. And you can't really tease that apart. So the question just goes unanswered. Having a very clear, while there is tremendous nuance within the signaling pathway and cell type specific and context, all that. I love the fact that now we have a tool where we're no longer in space about whether we're even doing what we think we're doing, technically from a tool standpoint. So yeah, it's no wonder that the Daily Group has been agonizing over making this tool for so long, because they just solved a major problem, not just for themselves, but for all of us. Thank you very much, George. You can send your wonderful ligand to the address in the notes. That's it for the roundup. We're about to talk to Mark Hatley, who certainly has struggled with Notch at some point in his life. We'll have to ask him about that. Before we get there, though, I have a quick message from STEMCELL Technologies. Are you studying mesenchymal stem cells? Well, whether you are or not, you might want to get a free wall chart that explains the functions, properties, and identifying markers of mesenchymal stem cells, so-called MSCs. To learn more, visit stemcell.com/MSC-properties-wall chart. All right, you guys, with us from St. Jude Children's Research Hospital, we have on the show associate member of St. Jude faculty, also director of the Division of Molecular Oncology there, Dr. Mark Hatley. The Hatley Lab's research focuses on the developmental and genetic mechanisms behind the cellular transformation in pediatric rhabdomyosarcoma. In the lab, they're working to understand the process of cancer transformation and how cell of origin dictates features of the disease. Mark, thank you so much for joining us on the show today.
Oh, thank you so much for the invitation. This is a spectacular opportunity.
Well, for us, it's an opportunity to talk to a unique guest amongst our typical cohort, or much more deeply focused in, garden variety stem cell biology, if that exists. Yeah, you're unique in that your primary focus is pediatric cancers, particularly, as I said, rhabdomyosarcoma. But the biology underlying your work with cancer is centered around transdifferentiation as a root cause of cancer emergence, which has a lot of overlap with the directed transdifferentiation or reprogramming that we explore pretty regularly on the show. Why don't we start with you giving us an overview of the work your group is focused on in your own words?
Yeah, sure. So I'm a pediatric oncologist and scientist at St. Jude, and my laboratory is really fundamentally trying to determine the intersection of normal development in children and how the programs are perturbed to give rise to pediatric cancer. Just 10,000 feet view, kids just aren't little adults and their diseases are quite unique. And when we think about how diseases occur in children, we really have to think about the moving target that we're treating. I mean, they're developing, everything is moving very fast. And that developmental process offers unique opportunities for things to go awry. And we're very interested in how different mutations, either genetic mutations or epigenetic changes, change the fate of cells that give rise to pediatric cancer. There's a cluster of pediatric malignancies called pediatric embryonal tumors. And these tumors, when we look under the microscope and dissect them genetically, they appear to be an arrested state of normal developmental processes. And so we're really trying to figure out how different cell states through development intersect with different genomic perturbations that give rise to pediatric cancer. And as you said in the intro, my laboratory uses rhabdomyosarcoma, which I think may have the distinction of having the longest name and in the freaking pediatric cancer, and I have to spell check that all the time myself. But so we use rhabdomyosarcoma as a model system to explore the intersection of the normal developmental processes and these genetic changes.
Yeah, thanks for the a little misspoke there. I said rhabdomyosarcoma, so thanks for bringing me back to center.
There you go. Thanks. Yeah, thanks for that. So we're talking kind of about how developmental programs can be hijacked pretty much, right, during the process of tumorigenesis. I mean, we learn so much from developmental biology, but this is a kind of a unique application in some ways. I mean, do you think we're actually approaching a point where DevBio might become such like a mainstream framework for understanding cancer, not just like pediatric tumors, but adult cancer as well?
Arun, that is the dream. And so that's exactly what we're trying to do. Having fundamental basis in developmental biology and becoming a pediatric oncologist, that's exactly what we're trying to explore. I think it's a very exciting time in developmental biology with iPS cells and directed differentiation, these gastroloids, and really doing in vitro human development. So we can now even step outside of the mouse models that have been the mainstay of my group and really start to use human model systems and the developmental processes and trajectories that we can manipulate in vitro to understand these connections.
Yeah, I don't know if you guys got that message from the NIH that they're really emphasizing the shift away from animal models, and so the human cell is taking center stage. I mean, it's convenient for me, but very disruptive for the field. But I think definitely the direction that everyone hopes to go in. As you said, Mark, this is the dream. Talk about dreams, maybe nightmares a little bit. I'm referring to about my postdoc, not the postdoc in whole, but there's so much frustration in any postdoc, and my frustration was with endothelium, right? That was my cellular medium. And working with endothelium that was derived from human embryonic stem cells. And whether we were differentiating from ESCs or isolating from primary tissues, the drift, as we called it, made it really, next to impossible, except if you were using cubic umbilical cord endothelium. It was really impossible to generate stable cultures. And one of the key facets of your own work is a focus on endothelium as a nightmare cell in terms of the cell of origin for rhabdomyosarcoma. And this is scary, right? Considering that most of the cells in your body are just a few cell diameters away from any capillary endothelial cell. But the biology underlying endothelial transformation to cancerous cells, right? Everyone talks about endo-MT, but the idea of transformation to cancer, that must be dramatically different from your garden variety endo-MT. Now, can you just elaborate on some of the nuance and the fascinating biology?
Sure. And so it was really quite a surprise when we found the connection between endothelial progenitor cells and this reprogramming event that shifted them into a muscle-like sarcoma called rhabdomyosarcoma. And so it was actually a funny story. So that we had a very similar paradigm that I thought was going to be the answer. So we started the exploration when I was a postdoc in Eric Olson's lab at UT Southwestern. And I went to Eric's lab. I met him like when I was a medical student. I was looking for pediatric residencies all across the country, looking for postdocs. I went and talked to Eric, and he wanted to make mouse models of rhabdomyosarcoma to better understand the disease. And I thought that was a perfect opportunity for me. And so I went to Eric's lab to do that. And I was doing everything that you would think to do using all of his muscle drivers, coupling it with RAS, oncogenic RAS, and deleting P53 and doing all the things that you normally would do, and not really getting the answers that we were expecting. And Eric's not a very patient man. And so that was a trying time, kind of like your endothelial, where you're into endothelial cells. And so, but there was this developmental biologist at UT Southwestern, John Graff, and he was very interested in adipose tissue. And he did a 2-component enhancer screen in Drosophila looking for things that suppressed fat body formation in the Drosophila larva. And he kept getting cubitus interruptus. And so, which is the Drosophila glee protein. And so then he thought, okay, this is cool. That's a signaling pathway that potentially I can modulate and block fat. Getting a skinny drug would be quite lucrative. So John went to 3T3L1 cells, which are a murine pre-adipocyte cell. And if he activated the hedgehog pathway in those cells, it blocked the ability of those cells to form mature adipose cells. And so he wanted to study this in vivo. So John wanted to take Andy McMahon's ROSA 26, lock, stop, lock, smoothen M2, which is an oncogenic, constitutively active smoothened allele, which activates the sonic hedgehog pathway. And he wanted to turn that on specifically in developing adipocytes. So he took this 5.4 kb promoter element called adipose protein 2. So the adipose protein 2, this promoter element had been studied for decades. Bruce Spiegelman did all these cat reporter assays in the early 90s, and people had done all this work showing that it wasn't in skeletal muscle, it was just fat, or so they thought. And so he used that promoter element to drive Kruger accommodase and wanted to activate the hedgehog pathway. So he did that, and the adipose in those mice, and those compound mutant mice, were completely normal. They had normal white fat, brown fat, beige fat, all the stuff. But all of the mice developed head neck rhabdomyosarcoma. And John wasn't interested in that. He knew what I was doing because benefits of being healthy. We used to work out in the gym together. And so he knew what I was doing. And so in collaboration with John and a pediatric pathologist, Renee Galindo at UT Southwestern, and Eric, we characterized this mouse model. The thing that was interesting to me was that it didn't appear to be coming from skeletal muscle. And that's really what we showed in the first paper, that we were getting the rhabdomyosarcoma in cells that weren't part of the normal muscle developmental trajectory. And that was cool because children get this tumor outside of skeletal muscle. So they can get tumors in the ovary, the cervix, the fat. They can get metastatic bone marrow disease without a primary tumor. So they occur in other places. And I thought this could be an insight to that. And so when I started my lab at St. Jude, I really wanted to explore what the cell of origin was. So to do that, we took this AP2 Cree that John had developed and just crossed it into the ROSA 26 tomato reporter mouse and asked what cells are being labeled. And sure enough, this Cree driver was not expressed in skeletal muscle. On myofibers are all not fluorescently red. And what we found was when we looked at the sections of the muscles where these tumors occur, which is in the neck, we looked at neck muscle and we saw that the muscle fibers were completely not labeled, but around the muscle fibers, they were studded with red stellate looking cells all around. And, at first you'd think, oh my God, these are satellite cells, muscle stem cells. But, there were too many. Those are only 4% of the nuclei in the muscle. And I mean, these were everywhere. And we're like, oh my God, what the hell? And so we went back and we did, some confocal and Z-stacks, and we found that these cells are actually not even in the muscle fibers. they're in the muscle interstitium. And so since we had them labeled with tomato, we used FACS, isolated those cells, did RNA-seq, and it seemed like these were endothelial cells. We went back to the mice, did co-staining with CD31 or PCAM, and all of the tomato labeled cells in the muscle of the mouse co-stained with CD31 and PCAM. So it seemed like these were endothelial cells. And so now we had this system where we had these cells labeled and we could follow their normal developmental trajectories. Then we went back and added the smoothened M2 where the mice get the tumors, and we were able to track when in development the proliferations occurred and where they were coming from. And so we were able to visualize the development of these tumors through the normal development of the mouse. And it tracks about the same timeline as you would expect in humans, trying to make, some evolutionary comparisons to time.
That's so cool. I love a deep dive like that. The whole, like the arc of your nascent career in 5 minutes. But I mean, really, that's so many years of work. But thanks for that synthesis. That's such a great story.
Yeah, and part of it spurred from a conversation in the gym. So I love
That's
that.
Right. That's right.
It's funny how things
stay healthy. Yeah.
There you go. You never know.
Yep.
So, kind of on that same topic of transdifferentiation or VCs into rhabdomyosarcoma under the right oncogenic conditions, like that's another important thing, right? It's got to be under the right conditions for this to happen. I'm wondering about how this might be able to inform other strategies for like downstream cancer diagnosis, detection, prevention, maybe even like in genetically predisposed populations. Is that something that you've looked to? And I guess one thing beyond that, is extending this to beyond sarcoma to like carcinoma or other types of cancers. Are these things that you guys have looked at?
So we haven't. So we've been looking specifically in rhabdomyosarcoma. I think some of these similar kind of vignettes are starting to come out in prostate cancer, in lung cancer, specifically small cell lung cancer, which is a neuroendocrine tumor. And so the thing about rhabdomyosarcoma is a terrible disease. We the chemotherapy that we use to treat it was published at St. Jude, 1971. The same year Richard Nixon declared war on cancer. The same year I was born, so it was nearly 54 years ago, and we have not made any progress in treating this disease. There are 350 kids diagnosed in the United States each year, and it's probably about at least 4 diseases that are all lumped together. One of the things that we've determined that can predict survival is location. So the location where the tumors occur in the kids really portends their likelihood to survive the treatment. When we think about that, but then couple it back to developmental biology, there's some very interesting connections. So all of our skeletal muscle is not the same. We usually think about muscle development as it comes from the paraxial mesoderm, from the somites. Somatic derived skeletal muscle really only explains muscle that derives from the shoulders down, like the body wall and the limbs. Your head and neck muscle is completely different. And 40% of the kids get rhabdomyosarcoma in the head and neck. So that the neck muscle derives from the branchial arches, from the cranial and the cranial mesoderm. So it's completely different origin. The specification transcription factors that turn on MyoD and MIF5, the key drivers of muscle fate, are completely different in the head and neck than they are in the somite. So PAX3 drives everything from the shoulders down. In the somite, in the head and neck, there's a constellation of factors. I think Eric had a paper before I had joined the lab that knocked out capsulin or TCF21 and myowar or musculin in mice, and they don't develop any of the facial muscles that are required for mastication and some of the pharyngeal muscles, but they make the rest of the muscles aren't perturbed. And those are some of the transcription factors that are required. And so what my lab is trying to do is now try to dissect how those cells of origin at different places, in combination with the genetic perturbations that we're seeing from these large scale genome efforts, like the Pediatric Cancer Genome Project that was formerly run by St. Jude, and how those things couple together to dictate the survival in these kids, because the location matters. more than just even the ability to resect the tumors and surgically take them out.
Yeah, this is such who would have thought? I didn't think that talking to pediatric cancer biologists that I'd get such a fun conversation that emphasized this kind of compartmentalization and modularity of developmental biology, right? I'm really visualizing the lineage tree of the human body here, which is so critical to our thinking, and everything we do as stem cell biologists and cancer biologists in your case, it's where let's walk this cell back in time. And the mechanisms that make up all these, modules of development are so unique. And here you focused on, of course, these many different facets of rhabdomyosarcoma, but your lab also focuses on like much more general concepts, right? I'm talking here about your work focusing on microRNA processing, specifically DICER, which has like a very wide purview, right? So, you've got the studies looking at the role of DICER mutations in or loss of function tumor formation, but the knock-on effect on downstream tumor suppressors, in all likelihood contributes, and the purview is so wide. So what's like the overlap in terms of mechanisms of oncogenic transformation that you see in your more kind of modular rhabdomyosarcoma and cell of origin work, be it endothelium or not, and then the more general DICER kind of mechanism pathway? Is there overlap? Are we talking about two kind of considerably different mechanisms?
There is overlap, and it actually goes to Arun's previous question about cancer predisposition. So the most common, as a pediatric oncologist, when you're having these horrible conversations with families, one of the things always they want to know is like, why did this happen? Like, what caused this cancer? Did my kid not eat enough broccoli? Did I do something, and in large part, there's no answer to that. And it's unfortunate that the answer is really just bad luck, but that's typically, what we're left with. The exception is these rare families that have germline mutations that predispose them to cancer, like Lee-Fermani syndrome, for example, or mutations in retinoblastoma, RB1. And so DICER1 is this key enzyme that's required in the processing of all microRNAs. And there's a cancer predisposition called DICER1 cancer predisposition, where families have a germline loss of one copy of DICER, and they're predisposed to get a multitude of different cancers, pleuropulmonary blastoma, which is a lung disease that kids get, as well as rhabdomyosarcoma. So we started we wanted to start exploring what the mechanism of that predisposition was. And we used our genetically engineered mouse models. And the second hit in DICER1 syndrome is very unique. It's typically not a loss of heterozygosity, so they don't lose the second allele. They actually get a missense mutation in the enzymatic domain that alters the function of the enzyme. So it shifts the balance of microRNA. So you have each microRNA makes a 5P microRNA and a 3P microRNA. If you get this mutation, you don't make any 5P microRNAs. And so you get no 5P, and then you get this excessive 3P. And so we, as coming out of Eric's lab and being a mouse person, I wanted to be very sophisticated and make an elegant mouse model. So I made a mouse model that would conditionally express that mutation, the missense mutation. And so we wanted to both knock out the normal DICER and replace it with the mutant DICER. That was where we were going. And so we did that. And so we made a DICER germline loss mouse that would recapitulate the background of these families. And we saw, just as others, Tyler Jacks, Christophe Marin, and Martine Rousseau here at St. Jude had all shown that loss of one copy of DICER was a haploinsufficient tumor suppressor. So just losing one copy, get tumors faster, more penetrant. It's, doing something, but it really wasn't clear what was happening. And so we wanted to be very cheeky, and we wanted to only focus on the tumor cell. So we took a conditional DICER allele with our model of this mutation where we wanted to replace it. and used that. So we're only in the tumor cell, we would delete DICER1 and then replace DICER1 with the mutant. And when we did that, we found that we had a huge surprise. So we were a haploinsufficient tumor suppressor and we had germline loss. But when we did it only in the tumor cell, we lost that haploinsufficiency. So this cancer predisposition was requiring cells other than the tumor cell, so that it was non-cell autonomous. So it wasn't just doing something to the tumor cell. There was some other cell involved. And so we spent now 15 years. I'm hoping that the story will be out soon. And so 15 years postdoc and a graduate student. I promised Randy Larson that it would be a 6-month project that turned into 5 years. So we went in a deep dive at trying to find those cells. And we found that those cells that if you lose one copy of DICER, the neutrophils massively expand in these tumors. So not only do we see mature neutrophils, we see immature neutrophils and even granulocyte monocyte progenitor cells, GMPs, which is really bizarre. So we're seeing the full spectrum of development of neutrophils in the tumor. And those neutrophils in the tumor are massively undergoing this process in this release of neutrophil extracellular traps, or undergoing this process called mitosis. And when they do that, these neutrophils are shooting out all these enzymes, metalloproteinases and other enzymes, and a ligand that actually activates mitogenic receptors on the tumor cell. And so we've kind of worked out that mechanism, blocked the process, rescued the phenotype, and actually have tested a drug that can block it, which rescues the phenotype. And so now we've identified an interesting mechanism in this cancer predisposition that may lead us to a therapy that can provide primary prevention for tumor formation in these families. And so I'm very excited about that, which made me neutrophils make endothelial cells look like cupcakes. Neutrophils are a hassle to work with. They live for hours and are very difficult. So I'm ready to get back to my endothelial cells.
Ready to get back to the roots. Yeah. No, that sounds good. I mean, you talked about these amazing model systems, these amazing mouse models that you've really used to drive the discoveries that you've made in your lab, to trace cell of origin, tumor evolution, all this kind of stuff, right? So we always love to talk about emerging tools on this podcast, and we've talked about single cell lineage tracing, spatial transcriptomics. How might those tools kind of further advance our understanding of pediatric cancer origins? And are there particular technologies or collaborations that you're really hyped about at St. Jude's that might enable some of the discoveries that you make?
Oh, absolutely. So one of the powerful things about this model system that we have is that the tumors are not arising from muscle cells. And since they're not arising from muscle cells, we can use this model to really dive in and figure out what the principal components are, what's establishing that tumor cell fate. And we can, since we have this lineage label, we can follow it temporally through development. So I have a graduate student in the lab, Grace Atkins, who is taking this AP2 Cre tomato lineage and following it from the time that the Cre recombinase gets expressed, turning on tomato, all the way through terminal differentiation of endothelial cells into adult muscle. So she's isolating cells by FACS and then doing multi-ohm, single cell multi-ohm sequencing so that we can look at the transcriptome as well as the chromatin accessibility within each cell. And we're defining the normal trajectory. And then she's also gone back and done that with smooth end. So we're seeing what happens with the activation of smooth end to determine how the cell fate, these muscle cells, gets established. And so that's been a really powerful tool. In the DICER story with the neutrophils and this non-cell autonomous interaction, spatial transcriptomics is going to be key to really dive into the, what is going on in the tumor microenvironment within our tumors. And then probably the coolest thing that we have going on, mice are great and it's been, what's got me to where I'm going, but I think I'm probably about as patient as Eric is, and mice take a long time. A lot of our models, we have recently gotten interested in this subset of rhabdomyosarcoma called alveolar rhabdomyosarcoma, which is driven by a chromosomal translocation 213, which generates an oncogenic fusion protein, PAX3-FOXO1. And those kids do terribly. They have some of the worst survival of any of the rhabdomyosarcoma patients. And it's very simple. They just have this one translocation. And if you look at the tumors, they have on average about 6 other missense mutations. So it's really that driver, the oncogenic fusion that's driving these tumors. And so about the same time that we're finding this link between endothelial cells and muscle and this what we call fusion negative rhabdomyosarcoma, are the ones driven by the smooth end, Margaret Buckingham, a developmental biologist in France, who's a goddess of muscle development, had identified a progenitor cell, this bipotent progenitor cell in the dermamyotome that can either can have fate, a myoblast fate or an endothelial fate. And the fate of that cell is determined by a transcriptional equilibrium between PAX3 and FOXC2. And so if it's PAX3, they become a myoblast, FOXC2, they become endothelium. And so that was kind of getting us this connection between PAX3 and endothelial cells. And PAX3 is one of the partners of the oncogenic fusion protein. And then Vincent Garcia and colleagues had taken human army alveolar rhabdomyosarcoma cells and performed virtual Hi-C on 4C-seq chromatin capture data. And what they identified is that this translocation, this 213 translocation, actually generated a very novel topological associated domain. And in that topological associated domain, the downstream or 3' cisregulatory region of FOXO1 was engaging the PAX3 promoter. And so that was really cool because they'd identified both a muscle enhancer and an endothelial enhancer in that cisregulatory region. So now there's this connection between potential and endothelial enhancer turning on this oncogenic fusion protein, which was interesting to us. This PAX3-FOXO1 also has pioneering activity, so it can open closed chromatin, so it could allow for accessibility of transcription factor binding sites that were outside of whatever lineage that the cells came from. And so we wanted to start to explore the mechanisms of this oncogenic fusion. And so we turned it on in endothelial cells using TEC-CRE. And so we compared it to the industry standard muscle-driven alveolar rhabdomyosarcoma model that was developed by Charles Keller when he was in Mario Capecchi's lab. And so all of our TEC-CRE PAX3-FOXO mice develop rhabdomyosarcoma, and we know they're coming from endothelial cells. We traced them, and then we took the muscle-derived cells, and we thought this is perfect because now we have an endothelial-derived tumor and we have a muscle-derived tumor. We can follow them. We know where they're coming from. And so if we take the tumors at the end, we can do something that we've never been able to do in the humans. We can actually try to determine if we can find any inkling, anything that gives us any indication of where the tumors came from. And for all the analysis we've done, we have not been able to determine which tumor came from which CRE driver. So we can't. So once this cell fate is determined, it's deterministic, and they lose the ability to dissect where they came from. And so that was cool. But that also means that we can't go back to the human and explore the origins with the information that we have currently. So we turned to human model systems. And so like Daylon said at the beginning, we turned to UVEC cells first because, they're the industry standard. So we took UVEC cells and did this experiment, kind of throwback to Bill Hahn, Chris Counter, and Bob Weinberg. And so we took those UVEC cells, immortalized them with T antigen and H-TERT and MYC, and which will not form tumors in mice. And then we added we transduced them with the lentivirus expressing this PAX3-FOXO1 fusion, and they transformed. They formed beautiful rhabdomyosarcoma in immunocompromised mice. And so now we've shown that these endothelial cells have the capacity to undergo this. The human endothelial cells have the same capacity to undergo this transformation, this transdifferentiation cell reprogramming event that the mice do. And so we wanted to have a little bit better capacity to test other lineages and be a little bit more pliable. So we wanted to use directed differentiation approaches using iPSCs. And so we took human iPSCs and then used an endothelial directed differentiation protocol from Chuck Murray, and then introduced the PAX3-FOXO1 expression with the lentivirus when those cells first became hemogenic mesoderm, which is around day 5 of the process. And those cells, too, also turn on MyoD and form this alveolar rhabdomyosarcoma when transplanted into immunocompromised mice. And so now we have this human system where we can really start to explore what exactly that oncofusion protein is doing. And so this allows us to determine, just like in the other mouse models, but now in a human system where we can determine what the principal components of determining what the cells are. So we can turn on the oncogenic fusion and then take samples temporally through time. And we really haven't been able to really deeply understand the tumor initiation process, which I think could help us not just better understand the disease, build better model systems that better recapitulate the kids' tumors, which could be better preclinical platforms, but maybe even identify new novel targets for therapy. And so right now we're using that system with a doxycycline inducible vector that allows us to turn on the oncogenic fusion at different time points through the trajectory. As well, we've added a degron tag to our oncogenic fusion so we can turn it on and turn it off, so we can toggle the expression of this oncogenic fusion both in vitro during the development or in mice in these implant models. So that's been really cool.
Yeah, I mean, the tech, right, has improved to such a high degree of resolution and sophistication that, yeah, I guess the end game now is seeing these things in real time. And
yeah,
that's a watershed, and it's happening to different degrees and different fields. But yeah, moving towards real time, single cell, or who knows, maybe moving forward, single molecule resolution, and in human systems, and breaking open black boxes, like in my field, implantation, right? Or in your case, these like fundamental cell transition initiation events that we couldn't really see. We could only see indirectly and infer. But the bottom line remains the same, right? Particularly in the case of childhood cancer, right? Because there's a high degree of cancers in the very young and the very old. And in the young, it's really kind of a continuation of this developmental process. So it makes a lot of sense in the overlap and alignment with developmental biologists and cancer researchers. But at St. Jude's, it's a really unique paradigm that focuses almost exclusively, right, on childhood disease and treatment. We've had a couple of folks from St. Jude's on the show before. Most recently, I think, was Shannon McKinney-Freeman. And the story of the founding, a true Hollywood narrative. Everyone should know this story. Look it up on Wikipedia. But the legacy is both inspiration as well as, I think, a template for practical altruism in medical care. Famously, patients are typically under 21 and aren't charged for their treatment. How does this core value make it a different place for you to practice science in medicine? And maybe, elaborate on other ways that you love working there.
Yeah. So as a pediatric oncologist, I'm in Disney World. I mean, this is St. Jude is a remarkable place. I mean, the mission, Danny Thomas established this institution, and we opened its doors in 1962 to treat children with catastrophic disease, regardless of their race, ethnicity, or belief systems, at no cost. And so when you're practicing medicine here and treating these kids with cancer, it's really remarkable to focus on that, and the families know that we've got them, and that there's not all these other things hanging about, like, how they're going to pay for this. They have insurance, they don't have insurance. We just do what's right, and take care of the kids. And it's really a remarkable way to practice medicine. The mission of St. Jude, is what I said, and it really is pervasive across everyone at the institution. I mean, everyone's on board. Like the people in the cafeteria, the people that come and pick up our biohazard waste, everyone's bought in. I mean, we're all on the same team. We have, generally you have one cafeteria, you're eating with families, everybody's together, and we're all centered on this one mission. And so it's been really remarkable. As well, when you think about like what happened, when St. Jude opened its doors, the cure for childhood leukemia, which is the most common tumor that children get, is acute lymphoblastic leukemia. The cure rate was 4%. I mean, it was ridiculous. And the first medical director, this man named Don Pinkel, was a real pioneer and a cowboy. And so he came here and did things that nobody else would do. So he just took a step back, figured out what the 4 biggest problems were in these kids and what was preventing their survival, and then developed what he called total therapy, which attacked every one of those components at every step of the treatment and provided this combination chemotherapy, radiation, and just people couldn't believe that kids were actually agreeing to do this. But they started curing kids. And that has been the foundation of what all of us are doing to treat ALL or acute lymphoblastic leukemia now. And now the survival is over 94%. Mm-hmm. Now that we have blinatumomab, it's probably going to get even better. Mm-hmm. And so, it's really remarkable. And that kind of esprit de corps and ability to challenge things is pervasive still today. And the way that we collaborate and just try to get things done.
Mm-hmm. Yeah, it's just an unbelievably special place with just an incredible mission. And as a new father of a couple kids, it's particularly powerful what you guys are doing. It really hits home. So thank you so much for sharing your story and your science with us, Mark. It's really great to catch up with you. And before we let you go here, we're going to ask you a couple of peripheral questions here. So first off, if you're not a scientist, what would you be?
Oh, that's a hard one. Because I don't think I work a day in my life right now. I love the lab. But if I wasn't a scientist, my dream job would be to be the director sportif of a Tour de France cycling team. I think there's so much parallels between that and science. It's all very process driven, analytical, attention to detail. There's this great clip of Lance Armstrong talking about the process of winning the Tour de France, and it really resonates. Tour de France isn't won in July; it's won in January and February.
I love that analogy. We've had a lot of folks here on the show who've had athletic backgrounds, and really it shows, that discipline, that hard work is common across both areas of interest, right? Science and sports too. So, wrapping things up, some rapid fire fill in the blanks here. When you're not conducting research, you are
on the bike.
There you go. I figured as much. If you could have one superpower, it would be
super speed.
Also, I feel like we're in a theme here, Mark.
Yeah, but that would be, just think about the number of mini preps and genotyping reactions you could do.
Fair enough. Or how fast you could go on the bike. Yeah. And finally, I can't start the day without
yoga.
Nice.
If I show up without yoga, my lab notices.
Wow. Stay centered, man. Well, I think it's doing some wonders, because you've got a great process. Listening to you talk about not just the history of your science, but also what you got cooking, and how that kind of aligns with your passion, which seems to be on a bike. It makes sense that you're doing what you're doing, and you're doing it so well. And thank you so much for sharing a lot of that with us and our listeners, Mark. This has been a real great chat.
Yeah. Thank you very much for the opportunity. I really appreciate it.
All right, y'all. That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at STEMCELL Podcast, Blue Sky at STEMCELLpodcast.com, or via email at info@stemcellpodcast.com with feedback or to suggest guests.